In the last 12 to 18 months, we've had some reports of bispecific T-cell engagers in the myeloma space. And basically the review from my presentation was that we're very excited from the early data from these trials. These are trials that we're doing in patients that have relapsed and refractory disease. And what I reviewed was that in these patients where they've had prior lines of therapy of 4, 6, or even 10 lines of prior therapy, this is the unmet medical need of myeloma when they have triple class refractory disease, when they're refractory to proteasome inhibitors and immunomodulatory drugs, and also the CD38 antibodies. We don't have a lot left in that space. And in fact, the average survival then is less than 12 months. So ASCO last year, Dr. Top presented the initial data from AMG 420. That was a continuous infusion bispecific T-cell engagers that bound to BCMA as well as binding to CD3 and activated the T-cells in the local myeloma environment. And what they showed last year at ASCO is that they did a phase 1 dose escalation trial. They showed that the maximum tolerated dose, or the MTD, was 400 micrograms per day. And in 10 patients that were treated at that level, they had a response rate of 70%. Now that's really dramatic. In other agents that we've tested in that space, the typical response range has been between maybe 20 and 35%. So to show a 70% response is pretty amazing, actually. Now, at ASH of last year, in December of 2019, we also had a presentation from BMS, and it was CC93269. And it was also a first-in-man. It was a bispecific drug. Now, this one was more IgG-like, meaning it had an FC component. One arm was a binder to BCMA. On the second arm, it actually had a second binder to BCMA and also a binder to CD3. So it was bivalent in that it bound to BCMA and CD3, but it had multiple binders to BCMA. And in that regard, they did a dose escalation study. And they found at the higher doses of this bispecific, and this was a step-up dosing from 6 milligrams to 10, or there were some patients that were just treated directly at 10. They reported on 9 patients, and Dr. Costas gave that presentation at ASH, and their response rate was 90%. It was pretty amazing. And I also showed a swimmer's plot from the BMS study showing that with follow-up in the order of 6 to 9 months, that there are a lot of patients who are continuing to be in response, and that actually responses appear to be deepening over time. So those 2 studies really got us quite excited about bispecifics. Now at ASCO this year, the Janssen basically put through their bispecific called teclistamab, and Dr. Usmani presented also a multi-institution phase 1 first-in-man trial. This was also an IgG-like bispecific, again binding to BCMA and CD3. And in this study, they actually had treated more patients. They treated about 78 patients from a very low dose, 0.3 micrograms per kilogram, up to a higher dose of 720 micrograms per kilogram. Now, we didn't have any data at the highest dose, at the 720 micrograms per kilogram, in terms of responses, but we had data at the 270. Now, when you look at bispecifics, there are some toxicities from bispecifics, and Dr. Osmani went over the bispecifics, and the bispecific toxicity from this drug, teclistamab. And there was some blood count suppression like you would see in a relapsed refractory myeloma population, but not so bad. Some neutropenia, some thrombocytopenia. And they had what we see on these kind of immune activating drugs, and that is cytokine release syndrome. So about a little over half of the patients of that 76 had cytokine release. It was about 60 to 70% cytokine release in the patient that received 38 micrograms per kilogram or more. And there was a small amount of neurotoxicity. I think it was 8% neurotoxicity, with only 1 patient having neurotoxicity that was grade 3 or higher. So actually very acceptable in terms of the toxicity profile. In terms of CRS, it was only grade 1 or 2. But the important part, and what I talked about, is where the response is. At 270 micrograms per kilogram, they had 12 patients, and the overall response rate was 67%. So an excellent response rate also. And again, they're still going up on their dose escalation. And then they showed us a swimmer's plot. And the swimmer's plot, I think it has us all very excited, because the swimmer's plot had patients out even as much as 19 months. And again, just like in the BMS trial, it showed that responses deepened over time, and they look to be durable. Now, we're all very excited to see patients being treated with these bispecifics having such amazing responses. But we really want to see a phase 2 study. We want to see a phase 2 study to see what the overall response is going to be at the, I'm going to call it the optimal biologic dose, whichever dose that they pick. And in my mind, that's going to be the dose that has the least amount of toxicity with the most amount of activity. And it may not be actually the highest dose tested. They may actually select a dose that's a little lower than highest because it'll have less side effects, less CRS per se, or less neurotoxicity, but still have a fair amount of activity. So we want to see a phase 2 trial at this optimal biologic dose, and we want to see what the PFS is. The other thing I did in my talk is I compared it to CAR T cells. And the early data from the CAR T cells, is showing that responses in the order of 60 to 100%, but a PFS of about 12 months. And the problem so far with the CAR data, and that's from BB2121, and also from data from the original study from the Legend trial in China, is that over time we're continuing to see relapses with patients treated with CAR Ts. And that's in stark contrast to patients that have non-Hodgkin's lymphoma. Because in non-Hodgkin's lymphoma, if they get a CD19 CAR and they're in remission for 3 to 6 months, they tend to be in remission ongoing. They don't see relapses really after 6 months. Where in myeloma, we have continued to see relapses at 6 months, at 12 months, at 18 months, even at out as much as 3 years later, we're having patients relapse. So it's really an interesting phenomenon in multiple myeloma, and very different than non-Hodgkin's lymphoma. So, but at the end of my talk, I said, we're very excited in the myeloma therapeutics that we have 3 different classes of drugs that are targeting BCMA. We have BCMA antibody drug conjugates, or ADCs, and the one that's actually soon probably to be approved is belantamab mafodotin, and that will be our first drug that we potentially could have to target BCMA and myeloma. And in that regard, that drug actually showed in these really refractory patients to have a response rate of about 30 to 35%. We're very excited on that. We're looking forward to using that drug. But the next ones are going to be the CAR T cells and the bispecifics. And those ones have even higher response rates, and we think will have more durability in terms of responses. But it won't be I don't think it'll be in 2020 that will have approval of any of those drugs. Those drugs, again, likely will be approved in 2021 or beyond. And so we're really excited. I showed my final slide is I showed what I believe is going to happen between 2025 and 2030. And really, I think that the CAR T cells, the bispecific T-cell engagers, these active antibody, active immunotherapy drugs are going to be used as part of smoldering myeloma, as part of newly diagnosed myeloma, as part of early relapse myeloma, and again in relapse refractory. And what it is we're going to find new targets. So if we go after BCMA and patients relapse with a BCMA negative tumor, we're going to have another target and another immunotherapeutic to give that patient. So I think that's really the future and where we're headed. So that's the end of my review from my bispecific review at ASCO.